So people will start joining now.
Yeah. Okay. Go. Good morning and welcome to VicNoTill's webinar with Nicole Masters. It's the first for us in the changing world to do it, and we found it a good way to get out some content. Firstly, I'd like to thank the AusGov Land Care programs for their support, Wimmera Catchment Management Authority, and VicNoTill's platinum sponsors, Hybrid Ag and Lorry Co, for their support and being able to put on events like this. Today we have with us agroecologist Nicole Masters, and in her own words, an international woman of mystery and soil siren, and Grant Sims, who Northern Victorian farmer, cropping cattle, and also running a cover crop seed business. I'm Tom Briggs, current board member with VicNoTill, farm in the northeast of Victoria. We run a cropping and sheep operation. We've been playing around with warm season covers for 4 years, disc and full stubber retention for 3 years, have taken out synthetic fertilizers, going to a foliar nutrition program, and also transitioning to a self-replacing ewe flock to build on the system. So that's a bit about us. Simsy, would you like to introduce yourself and a bit about what your system is and where you've transitioned to and from over the years?
Oh, yep. So yeah, I was, I suppose, been on the board with VicNoTill for the last 5 years and off it now. But yeah, we're predominantly cropping, but running cattle as well and multi-species covers, forage crops. I suppose in I think it was 2008, we did away with MAP and fungicides, insecticides, and since then I've been using a foliar liquid fertilizer, like a biologically made one put together. Yeah, and just always learning and evolving. We do a lot of on-farm trials and to work out a lot of stuff ourselves. Yeah, haven't used seed or conventional seed dressings as well for 10 years. We've got some irrigation country, and you know, it's good. It's interesting to see we're still able to grow high yielding crops, you know, with water on irrigation without the need for these high synthetic inputs and you know, fungicides, insecticides. So yeah, we've seen, you know, it takes time to build your soil and to get things functioning, but I think it's very exciting if we can get life back into the soil, biology and nutritional balance, you know, what the soil's able to achieve. And we're still not where we want to get to, but we're always trying to keep improving things.
Yeah. Like everyone on a continual curve.
Yep.
Yeah. All right. Righto. So Nicole, any sort of, out of what Grant and I are doing, any similarities you'd see or like to discuss about what we're currently doing?
I think we asked everybody to send in questions. So we've had some really interesting questions, and we've got a whole 2 pages of them. And a lot of those questions are really technical and thinking about either nutrient, insecticidal, fungicidal interactions with either biology and mycorrhizae or plant production. So I think what I'd really like to explore is some of the reasoning behind why you do some of the practices that you do. Maybe it's been some of the learning and some of the decisions maybe that with hindsight you might have changed. And I'd like to explore that a little bit. Yeah, I'm always on the road, and so, you know, it's always interesting just to see what people are having success with and where they are doing the different things that they are doing. I'm getting a lot of questions about seed treatment. I actually think I need to make a video of making your own biological type seed treatments. So I wouldn't mind, like hearing a little bit from you guys about why have you pulled the insecticides? What have been the cost or not of doing something like that? So yeah, I wouldn't mind exploring that with you.
Yeah, I suppose on what we've taken out of the system, probably one of the easiest has actually been the insecticides. Pulling them out hasn't been an issue. We've got a bit of an issue at the moment with some long season canola. We got some armyworm going through it. We've had warm conditions and rain, and there's just anything that wants to eat anything's showing up. But other than this moment and trying to get a canola established, insecticides, dropping them out, and fungicides has probably been actually one of the easiest things to do. The challenge more so has been around crop nutrition, and also what chemicals you do and don't use around herbicides for weed control, and having weed control has probably been, for us, the biggest sort of learning curve. Nutrition and herbicides, what to keep in, what to keep out. We've seen periods of high insect pressure was it 2018 when crops got frosted everywhere and everyone went and cut their canola for hay, and we one of the only canola crops left in the area, so we had every single aphid in the district come and hit our canola, and it was a tree of aphids. Walked out there, you saw beneficials. A week later you would have sworn that you'd gone through with an insecticide. So it's also a period of having the stomach and knowing what numbers and thresholds are to not go and put that insecticide out. But it's also been one where once you know what you're looking for, it's actually been probably one of the easiest things. Other than establishing canola crops, that's just a continual headache.
Yeah, and I think the question is around some of the choice of crops that we plant and we use. And, is this the right growing condition for this crop? There's been a lot of interesting changes in terms of seed selection and what the seed guys are doing, and that a lot of these crops don't have their microbial relationships anymore. They say since 1975, wheat isn't even an obligate mycorrhizal plant anymore, so that's happened through breeding. And so what have we done to canola? So canola will have biological companions, and if we've disrupted that biological community, those seeds are bred for high input systems, lots of insecticides and pesticides, and then we try and put them into a regenerative system. And when I first started out, I was working in the apple industry and working with organic producers, and just finding those big conventional huge production crops just didn't have biological communication really happening. It's like those plants were kind of handicapped and they needed you to kind of feed them and put all this junk on. And so I wonder if there's an opportunity for regenerative producers to actually be looking at producing seed lines that are plants that have reconnected. And I've talked to some seed companies about it, and they are starting to have this conversation themselves. And I guess, Grant, now that you've got this business that you're developing, actually having seed that you are producing year on your place, what is the opportunity for that?
Yeah, definitely. I think, and I know talking around some mates that have, I suppose, transition and change systems, and they're seeing, in the same paddock under the same management, different varieties of wheat responding, or you could say some are sooks and some will adapt to the system. So I think there's a real place for some of these trials to look at root systems and how they can connect. And I suppose one of the important things that we work on and think about is the fungi component. It's something that's, I think, sometimes forgotten about, because a lot of this, either collaboration or sharing of nutrients or disease protection and all that, comes back to fungal associations. And if we're, a lot of these trial sites are doing tillage or fungicides either on the seed or throughout the cropping year, it's hard to get that sort of rolling. And the other thing, just pointing out on the canola, we see it in the, multi-species. We don't plant any monoculture canola anymore. It's always got a companion, but it always sort of, I find interesting that if you go to a paddock where you had canola last year, you get some rain early, you get volunteer canola popping up with no seed treatment on it, no fertilizer, no nothing. It always powers away into a cabbaging plant and seems to grow really well. As soon as you buy the seed with the seed dressing on it and put all your, the right best practice fertilizer under it, it's like a beacon. We seem to get hammered all the time with it's the most susceptible probably plant, the brassicas, to anything. And it just always sort of find it interesting. The volunteers seem to not have an issue as much.
Yeah, and I think I read a really interesting letter that was written by the USDA talking about those seed treatments, and their conclusion was that for every one benefit, there were 99 costs to those seed treatments. And the conclusion was, why are we even using these seed treatments? And that's coming from a government level. And it's like some of the research around, those neonicotinoids change gene expression in the plant. They change 600 genes in the actual plant. So you're basically, and some of those genes are for insect resistance and cell wall strength and disease resistance. And so, I mean, it's a great sales pitch. We're going to put this seed dressing on, it's going to do this in the plant, and now you're going to need a fungicide. But I think we're handicapping those plants. And I think the sooner we can kind of get some of this stuff out and then really look at biological seed treatments. So using things like a trichoderma or a mycorrhizae. Guys are using bacillus. Luke Harrington's was saying he's using molasses and liquid vermicast. Really setting that plant up for success, rather than, okay, we're going to give it a prophylactic, and that actually prophylactic is going to create more problems further down the track. Are you guys using seed treatments?
Other than the long season canola that we bought this year, we haven't used a seed treatment for three years. We've been putting biologicals on it, and this year we're putting a few more trace elements, but fungicides, no, we've been planting basically bare seed for three years.
Yeah, sorry, I meant biological seed treatments. So you are using
putting a worm vermicast on it. No compost extract. We haven't been doing compost extracts. It's one thing I'd like to get into one day, but it's just got other things on the list at the moment. But worm extract on it, worm vermicast, sorry.
Yeah. For you, Grant?
Yeah, we've only, if we're buying a new lines of canola, comes, already treated with the conventional one. But we haven't used it for, over 10 or 11 years, on all our other crops. We've been playing around with lots of different things as seed, and we're still looking at perfecting, a sort of a good seed inoculant or brew. But we've done the worm casting extracts. That's pretty easy. And then last year we did a lot of the Pseudomonas, Bacillus and Trichoderma, and had that as a live culture and put that on the seed. And that was pretty, seemed to work really well. We had good sort of the dreadlocks going on the roots, and we sort of had low input such as nitrogen, phosphorus, or synthetic forms of that up front, but had canopy closure very quick, and so that seemed to be pretty promising. I think we could that trichoderma, and you can sort of lift on that, Nicole. It's for disease protection and things on the plant, getting that rhizosphere or the good biology in there to sort of get all those relationships going and set the plant up to defend itself, rather than some of these. I talked to one of the barley breeders last year, and some of the seeds they're saying you've got to factor in a fungicide if you're going to run with this line. It just factor it in as part of the budget. It's a good yielder, but it won't be able to grow without at least one fungicide. So I think that's a bit of a concern.
Yeah, and again, a bit of a fallacy in a sales pitch. There's a couple questions here, and I think we've got questions that we were asked in the last week, but I think it's keep that kind of conversation flowing, and I think these questions kind of feed into the questions that we've had so far. So there's a question here about, is there a way to increase mycorrhizal interaction with newer bread crops? Yeah, I think with the newer bread crops, it is quite fascinating because you look at what is happening with those root systems. And I was trying to find a photograph of, I took a photograph of kamut next to wheat. So kamut is that ancient grain that could actually be used as a wheat substitute. I think I've talked to you about this, Grant. I'd really like to see people in Australia growing it, Corazon wheat. But anyway, to look at the rooting systems, I mean, they had these massive root systems with all of that Rastafarian or the rhizosheath roots that we've been talking about, compared to this wheat plant, which had these pathetic little roots, and the roots were quite clean. And it's like, we've done something in that hybridization of just trying to chase purely yield. I mean, the kamut itself still yields well, but you know, it's the same with like breeding sheep or whatever, and you're going to select just for reproduction and you lose feet or whatever you're doing. But
so if we buy a new wheat variety or barley variety, and at initial purchase it's come out of an intensive system, can you breed and you retain your seed? Can you breed a mycorrhizal association, like a plant that's more like, will it change? Is that possible through epigenetic of retaining the seed, even though it might start off from a low base?
Yeah, and then we're getting into that epigenetics concept, which is that we can change. Actually, I had a couple of slides. If you don't mind, this might be a good time to kind of talk about it. Just thinking about what is happening in the seed and how is it that we can influence stuff like that. And so if we think about the seed microbiome, so can you see my screen?
Yep.
Right. So there is actually a seed microbiome, which is really new, cutting edge research. Like, people didn't really think, oh, you mean there's microbiology inside a seed? And it's like, well, yeah, of course there is. And so that microbiome comes from either vertical, which means that it's coming from that mother plant as such, or horizontal, which means it's coming from the air and soil and the environment around it. So that if you're thinking of a conventional cropping system where they're growing seed, this microbiome from the start is compromised because you're not going to have that diversity. And what you find is some of those organisms that are in those environments are your pathogens, right? So there's actually diseases that are inside the seed waiting, which is a bit of a worry.
Wow.
And so I think, so this is looking at where does the seed microbiome come from. It's coming from within the actual roots and the stem. So there's microbiology that are inside the xylem of the plant, or inside like an endophyte living inside plants. And it's part of what plants are doing for nutrition and for health. But, you know, there's this whole world of microbiology that's working away inside plants. And so the seed microbiome, why we want to have a diversity of microorganisms, is it's going to really help to prime that plant and speed up germination. And here's an image of those Rastafarian roots that Grant was referring to. These microbiology then help to induce that plant defense system. So say you did have sclerotinia or rhizoctonia or something, having that seed microbiome already set up enables it to defend itself. It also enables it to solubilize nutrients. So it's helping with nutrient uptake and nitrogen fixation. They are producing plant hormones. So that's going to help with rooting length, shoot length. And like I said, there can actually be pathogens in here. One of the processes that's happening, and this is fascinating, is that 30 to 100% of nutrients that the seedling absorbs comes from absorbing bacteria at that root tip. So that very moment that that seedling starts to emerge, it's feeding bacteria around that root zone, and then it's absorbing that bacteria. It blows open the membrane on the outside of that bacteria's body and absorbs those nutrients. So it's not having a package of phosphate out there or some kind of nutrients. It's actually having microbiology ready to go, stimulated and responding to the plant. And the other thing that's quite interesting is that microbiology that's in that rhizophagy process also is absorbed by the plant and initiates root hairs. So if you have a look and dig up some of your, so you got a new variety, go and have a look, and what you'll find often is there's not a lot of root hairs, and those roots are really, really clean. And it's indicating to you like how much microbiology is really, really functional. And so we want to see loads and loads of hairs. So if you look at that image on the left, like the wild type, which is a sunflower, we want to see lots and lots of hairs. We want to see soil stuck to it, because that's your disease protection. That's where nutrient and water uptake is coming in. And so we find this is being reduced through what they're doing with breeding, by not having that microbiology in that seed or around the seed coating. And so we see less disease protection, nutrient and water uptake right from the moment that that plant's starting to germinate. So if I'm thinking about, I'm wanting to encourage mycorrhizae, I want to have all that microbiology, I'm never, ever, ever going to use neonicotinoids on the seed, right? They do disrupt your mycorrhizae, they do disrupt all of that system in the plant. And then what you guys are doing is doing things like coating seeds with compost or vermicast extracts. Again, you want to get some really good quality extracts, and there are these commercial biologicals like your trichoderma, your mycorrhizae, your azotobacter, like your nitrogen fixers, and actually getting them onto the seed. I do prefer the worm extracts because they have such diversity in them. You're not having to kind of package it and guess what's in it. And then either dripping that down the seed, down, which I think you're doing, Grant, putting it down when you're planting. We're also putting dried vermicast down the drill in the seed box. And so we want to see, and this is a photo from the Haggertys in Western Australia, but you want to see that rhizosheath development from the moment that that plant gets out, because the rhizosheath is making a huge difference in terms of a buffer to temperature or pH. We're seeing a lot of aluminium problems in Australia and in New Zealand on ash soils. If you can have a plant that's germinating and creating that rhizosheath from the minute that it's getting out in the world, it's going to start to be able to protect itself. And that pH can be as much as 2 units difference. So that's kind of a priority for thinking about what are we doing in terms of looking after those seeds. So have we got systems like that? And you hear a lot of people coming forth with ideas like it's new. This stuff isn't new, right? These ideas of doing seed treatments, like you look at the Korean natural farming. So one of their seed treatment mixes, basically they recommend putting 5 kilos of a mature compost, mixing that with a bit of molasses and a cup of milk and making a slurry. And it's going to be thick enough to coat the seed, but not so thick that it will pour off. So we're seeing Johnson Sue, so the Johnson Sue compost, David Johnson, he's talking about taking ½ liter of a compost slurry, so that material, mixing it with 10 kilos of seed, and then adding that into 120 kilos. So the idea of mixing it like that is so you don't get all clumpy, and then he's drying it. I just think on large scale operations that can be really time consuming. So instead actually applying directly to your seed before you dry. Yeah, so I'll finish now, but I just wanted to share that while people were asking these questions.
I'll just add in there, Nicole, on some of the just practical stuff, getting these seed inoculants and treatments on. And firstly, also, I suppose, with the breeding of new lines and that, because I don't want to, we have NVT wheat variety trials on our farm every year. So we see a lot of new varieties coming out. And I just sort of need a little disclaimer. I think they're doing a great job. Generally, a lot of these, because I mean, some of the new varieties, like we used a new variety of barley last year against one of our old that we've retained for years, and it smashed it in yield in our system. So not all of them are, there's a lot of good work getting done there, I suppose, as well. And that was in our, with low input, or so just sort of putting that out there. But applying I think some of these seed treatments, like you've just mentioned there, is something we can all do ourselves on farm. And with every type of agriculture at the moment, cost of production is a thing that we've all got to watch, and some of these things we can really reduce our input costs, and it sets the plan up from a young age so that it doesn't become so dependent on all the other inputs later on, like the fungicides, insecticides, and also helping it make its own phosphorus and nitrogen available and things like that. For a practical part of applying, we've gone up to 12 litres per tonne of whatever the liquid, and that's about the max, I reckon. And the grain starts heaping up and getting a bit wet. then we just got to make sure that we leave it in the truck. By the time we get it to the truck and into the seeder, it's generally dried out enough, but you've got to watch, because when you're calibrating, you know, it'll run differently if it's wetter or drier. But a lot of times with 5 litres a tonne is pretty easy to put on.
Yeah.
Yeah.
Do you do it up an auger, or do you have a mixing wagon? We have an old PDO hammer mill that we use, that we bypass the hammers into the additives, put a ton in, and then put 10 litres of product on, and then some dry product in there as well. Are you doing it up an auger out of a silo or in a mixer? How are you applying it, Grant?
Yeah, we do it up an auger because we're doing large bigger tons, I suppose. Like we might do 10 or 15 tonne at a time going into the truck, because then that'll go out and we'll sow that out in a 24-hour period. So in the mixers is too slow and mucking around when we're trying to do bigger areas. But yeah, up the auger, we drill a couple of holes in the auger and put 2 nozzles, like a fan, you know, and just calibrate have a little 12-volt sprayer, so we get a certain amount of litres per minute and then know how many tonnes a minute we do. And it puts it on really, really well.
Yeah.
So there's a question here about how do you know if a product is good quality? So I know for myself, when I used to actually have my hands in the ground, is that I would do so many different things that it would be hard to pull out and go, well, I know that was working quite well, because I mean, you're doing trace elements, you're doing seed treatments, biologicals. So how do you guys figure out whether or not something's a good product?
I suppose part of it's also been the learning curve. When you're starting to put all different sorts of bits and pieces on the seed, it's hard to pinpoint one that either does nothing or detracts. So we're using a bought liquid vermicast off Nutrisoil on the seed. Believe it to be high quality. And then as far as trace elements, we've just been sort of learning about what's been showing up in tissue tests, our own observations. So not making our own, but buying off the shelf products and putting on the seed. But that's just been a learning curve of, we think we need to add this in, or I don't think that did anything for us. So when you're starting to get multiples, how many trials can you have in one paddock of trying this against this against this? So some of it's just the learning curve of what we think is needed or hasn't done or provided any benefit.
For you, Grant?
Yeah, that's a good question, I suppose, because it is tricky when you got a whole looking at a system. It's hard to narrow down one thing or one input, and sometimes some of these things might take time. Like it might be a couple of years to start to build these things in. There's certain products you can foliar apply or put in and see whether you've got some sort of response. I think it's always important to leave, and a lot of times when you're in the heat of sowing, you just do the whole paddock and it's just easy. But leaving test strips is very good, because that's you've got to be able to have something to quantify or measure. The Briggs, that's probably a simple, easy one. You can spray some of these things on, test the plant for Briggs and then spray it on, test it an hour later or two, and at the end of the day, if we're getting a really positive lift in the Briggs, well, that's good. So there's a few simple things we can do in the field on the run to check some of these things.
Yeah, we had an interesting scenario last season. I think we put on too much worm liquid, so the Bricks in the lucerne went from 13 to 10 and stayed 13 in the control, but a week later it was up at 20. So it was like, I think we just overloaded the system. So I think it's worth testing it within 40 minutes if you're trialing some of these different things, like the different fish or people are asking. And then test it within 40 minutes and then come back a week later and just see, actually, am I putting too much on, and just see, because some of these inputs we're finding, there's a suppression if you're going too high, and that could be a suppression at 1/2 litre. And so you need to figure that out. At what point is that sweet spot of, we don't want to actually shut systems down, because especially, like, I think you guys get your system starting to really, really work for you. It doesn't want you to keep fiddling and adding stuff in.
Yeah. Which is, I suppose, where the plant tissue, the tissue testing or SAP testing plays a part in not actually getting a response from the plant, seeing what it's actually needing, and rather than gut feel, I've just got to put something on.
Yeah.
Plays a big part, not overloading it one way or the other, but still it's, you go and get a SAP test or a tissue test, and then you talk to someone, how much of this should I put on? Oh, we don't know. We've never done anything like this before. So you got to sort of feel it out yourself as well as to what you think's an appropriate amount and action to take.
Yeah. Yeah. And that's why I think it's helpful if you're working alongside these companies, if they are doing some of that testing in the field. They have actually got some trial results. We saw interesting stuff in New Zealand with mycorrhizae, with 7 of the leading mycorrhizal providers who were selling. They got tested and none of them were viable. Like, these are the 7 biggest companies and none of those mycorrhizal products were viable. So asking for viability tests when you're dealing with microbiology, and testing for yourself.
Just notice there's a few questions coming in on that. How do you tell if a product's good or not? Is that a way to get them to send you an analysis they should be able to supply an analysis pre-sale of their product?
Yeah, definitely. I mean, especially around biologicals, to ask for that activity. I'm just seeing some companies kind of sneaky little things in, like they might be putting hormones in to get a growth response, and the only way you're going to figure that out is to do that for yourself, because hormones will drop the Briggs, and if your calcium is low, like, you know, below 60%, you'll see that with the use of hormonal products, your calcium starts to, like, disappear quite fast, we see. So that, I mean, I do think using the refractometer can be one of your better tools, plus doing your leaf tissue test and digging holes. Someone's asked about ways to increase mycorrhizal interactions. I think you want to figure out, why is mycorrhizae not working as well as it should? So really using your diagnostics. Is it something that you're doing? Maybe it is something to do with breeding. Are you adding on too much phosphate? And this is a question that I think, Tom, that you might have printed out from yesterday, is like, at what point do we see mycorrhizae shut down? So mycorrhizae really, you know, the plant's like feeding it in return for, you know, the phosphorus and zinc and other nutrients. And if it gets, if you have rapidly available soluble P, the plant is not going to invest in that mycorrhizae. And that level is going to depend on the type of phosphate, so how rapidly available it is, and on soil types and on temperature and all sorts of things. So you need to do this testing for yourself, and you know, and looking at colonization in that early phase, you know, when plants are like 3 weeks old, send off a mycorrhizal test and see, you know, what percentage colonization have you got, and try and use your diagnostics before I'd be reaching for a packaged product necessarily. What were we going to say, Grant?
Oh no, well, it's just there's one question there also, like with the mycorrhizae linking up, you know, what levels, you know, if we are applying some of these synthetic phosphorus and DAP and urea and things like that, Matt, as to what point, you know, the question here is what point, how high before it starts doing damage to that. And then I suppose another thing that we've been told is applying the carbon or the humate to it to help buffer that. So if you want to elaborate on that, what levels become detrimental, I suppose.
Yeah. We know that 50 kilos of MAP by itself is going to shut that mycorrhizae down. When you look at the research, they talk about parts per million in terms of soluble availability, and that's really hard for you to figure that out, because it's going to depend on soil types and stuff, and that's where it comes to probably testing your own mycorrhizae. But I think certainly less is more in this case, and then looking at how do you increase the efficacy? So it could be your humates, or it could be your fish hydrolysates. So those fish hydrolysates they make with a phosphoric acid, and then you've got all this carbon with it. Some work done by New Zealand researchers showed that 1 unit of P in fish gave the equivalent increase in dry matter and phosphate response in the plant to 180 units of superphosphate. So 1 unit to 180 units. So we're not talking like for like. And so trying to figure that out for yourself is where do we see that cut off?
Was that fish hydrolysis, was that applied on seed or foliar?
Yeah, you can do a soil drench, you can do it foliar, you can put it down. It has a pH of 3.5, so you're going to be diluting that and put it in with you. Yeah, put it in.
We liquid inject fish and humates into the soil as a food source, and it's a really good fungi food source. Find that very, very good. And I suppose last year was interesting. We were sowing and ran out of. We were using some guano last year and some of our biological liquid fertilizers, and it didn't come on the weekend, so we just went into town and got some MAP and just kept going. And it was interesting to see. I think we've got to get used to visually looking like the crops. We had way more early vigor, more biomass through the winter with the MAP, and I was thinking, geez, what are we're missing out here? But then as we got into the spring, the MAP all just shut down and went yellow, started turning. We had a lot more disease in it. It was coming in like leaf disease and things like that. And that where we'd done the more biological things, they just kept green, kept going and kept going. And in the end, I think the yield was exactly the same, but we were still in the game a lot longer with the other ones. We didn't get a late rain, but if we had have, the plant's still green, ready to we're still in the game. So I think, talking about MAP and how quick it's really good early. You get everything a flush of it, so you get way more early vigor and biomass early, but then it can potentially lock up and you run out at the end. I don't know if you want to comment on any of that, Nicole.
Yeah, well, no, I think that's exactly what we see in the field is that it locks up. Any of those soluble forms are going to lock up really fast, and they're also suppressing the organisms that you want working for you to help release that phosphorus. So really the aim of the game is how do you get those guys working for you instead of trying to drip feed stuff all the time, and that's where your leaf tests come in and go, do I have adequate phosphorus? Do I need to really be pushing this? And I really like the idea of putting fish down with the seed and using that as your early, cooler season phosphorus bump.
What about like we're finding, companions, multi-species, things like that, like clovers that have found their root exudates can break the calcium phosphorus bond in the soil and things like that, buckwheat, other plants that, and we've seen and played around with it can really help stimulate that phosphorus availability or other nutrients.
I don't know if you've had a look at Lauren Steinledge. Did you meet him, Grant, when you went in Iowa?
I know Lauren.
Yeah, I mean that idea of like having a continual living, growing crop that you're planting into. So he's using white clover, for instance, or having cover crop mixes between corn, and seeing those kind of responses that those plants are releasing phosphorus. I mean, all of your legumes except lupins are, and lupin's got another role, right? But all those legumes are big fungal feeders in terms of how do we get more fungal diversity in there. Lupins, like your buckwheat, have an adventitious root system that releases really powerful acids to break that bond again. So it's why, we use them in gardens or whatever to do your intercropping. But I think the people are really cracking this intercropping or the piolas or, having more diversity in that system, because those different plants are then relating to different types of microbiology as well to help release these bound nutrients. So we can really get off the nutrient treadmill.
Yeah.
Yep.
There's a question about putting trace elements with liquid. Saying if you put trace elements with liquid vermicast, you can cut it back 10 to 20%. There's actually fulvic and humic acids in liquid vermicast, so they help to increase cell wall permeability. So you could actually not only drop by 30%, you can do more than that. So I don't know, Grant, are you making your own trace elements?
Yep. Yeah,
And so are you finding
pardon?
you're dropping right down?
Yeah. So, yeah, we I don't know, for years have sort of had a product that's got all trace elements and the main ones, and now we're making our own. But yeah, and that's we've seen on our SAP tests, you know, back years ago, and we're just on straight MAP. You would have deficiencies in whatever it may be, copper, zinc, or whatever. Now they're coming more balanced, and I think that all helps with their, you know, keeping our Briggs level up, you know, keeping disease and insects at bay. And so, yeah, I think like when we're over in New Zealand with yourself there, Nicole, they're saying, you know, add microbes to the minerals, you know, and then humates. So if we get that biology in with the minerals, everything seems to amplify each other.
Yeah. So we're looking at what traditionally might be between 1 to 10 kilos of a trace element, now down to like 100 grams of a trace element, and seeing the same response in the plant. And brewing that through fulvic acid or using compost, by making a compost extract as such, and then adding trace elements into that to get it to chelate in a manner that you still have the biological metabolites there, but seeing just massive improvements in terms of efficiency of those trace elements.
Is there much known around with the use of humates and being able to drop and gaining efficiency with the use of it? Is there as much, is it as well known around the herbicide use with using fulvics and dropping it, or is that a bit more of an experimental area? With the glyphosate shortage here with the coronavirus, we were going as minimal rates as we could, and we're adding fulvic, and we were dropping less than 30% for the amount of active constituent to go out, and we did not see a kill job. But is that something where it's not as well known whether the fulvics enhance the job of it, but you still get the efficacy? Where is the humates nutrient wise, that's more of a known that if you drop it, you add it in, add that source, that it increases the efficiency?
Yeah, the work that was done on that cell wall permeability was on humics. That fulvic, is going to increase efficacy. So you're saying you dropped by 30%, or you dropped it down?
Wasn't, we had as much product as we had on hand. We worked out how much area we're trying to cover, and then added the fulvic in, and it just happened to be a bit, it was about 25% drop in what we would have used rate wise. And it just, it wasn't enough. We didn't see the similar sort of job. One paddock, we did a trial paddock where we didn't put fulvic in, and we had the recommended rate, and we got a good kill. But where we dropped rate, we didn't get as good a kill.
And so what was the pH in your tank?
You're asking questions I don't know now. It was rainwater. It was out of a rainwater tank. What that actually is, I don't know off the top of my head.
Yeah, so I would get a pH meter. Everyone that's using herbicides or foliars, get a pH meter. They're not very expensive, and start to look at, because, a lot of those chemicals are more, the efficacy comes down to, between 3.5 and 4.5, 4.6. And so we're finding, if you're not getting that pH down, and fulvic acid, so humic acid's alkaline and fulvic acid's acid. I'd be really curious about that. We're not having that problem. We're putting fulvic acid in and finding good kills. So
this year on our knockdowns, we used the product with the fulvic and molasses in the glyphosate, and we had total, and I think it was like 1.2 litres a hectare of glyphosate at like 450, a low, a cheaper sort of one, and we had complete brownout in 5 to 7 days. Like, it's working really good. So I think some of these things can help. And I know there was a question in there somewhere on, looking at when we're growing our monoculture grain crops, I suppose, and there's, you know, ryegrass and things like that, and what chemicals or herbicides we can use to control that, and you know, detrimental to soil biology. I think, have you got that question there, Tom? And then whether you could use some of these things to buffer, or there's better choices or rotations, or
Yeah. So if we got, well, for us, probably our biggest headache here is ryegrass, and whether we're better off taking action with a pre-emergent herbicide to try get control, but then buffer against those early actions to get crop established. But then what we can do in crop to mitigate against that early practice, whether that's too detrimental early on, to then remediate some of those early actions, whether we're better off taking the early action and establishing a healthy crop, and then in crop remediating that early action. That's at home here, that's probably our biggest issue, is around ryegrass. It's so suppressive. It takes some hard chemistry to get on top of it, but if we don't, then our crops, you know, it's also how long does it take for our system to evolve where ryegrass isn't an issue and it's not so competitive. But in the early stages when it is, do we have to take that early action and then drive crop performance and inputs to remediate against that early action?
Oh, I'd say, like, on some of that, because I get asked that a lot with, you know, using chemicals on the ryegrass and things like that. At the end of the day, you know, we've got to we can't be planning into those conditions, because it's gonna be detrimental to yield and the outcome. But I suppose you've got to ask yourself, some of the things we're doing, are they creating the environment or the conditions for those ryegrasses to thrive in? And you know, we see on some soil types, you know, it'll grow to nearly a line, and it's not, you know, you put the same chemistry over everything, but it's, you know, maybe looking back at what's going on, you know, in the soil, and, you know, changing the dynamics of the soil so that it's not as favourable as for those plants to thrive in, I suppose, whether through rotations or making them more fungal or whatever.
I just happened to notice last year we were still a low rainfall year here last year, so we didn't have waterlogging, which is usually one of our biggest issues. A wet drought is more of an issue than a dry drought here, and was noticing, when you know your paddocks long enough, you know where your wet spots are. You can nearly walk to them with your eyes closed in a paddock and land in the spot from the gate. And last harvest was noticing crop going from yielding to very little, and where ryegrass had become dominant. And when we're shifting bins across the paddock, I walked across with a shovel, and I'd drop the shovel in where there was healthy crop and there was nice porosity in the soil. Then where you'd get into spots where the ryegrass was either completely dominant or there was some crop, but mainly ryegrass, it'd go to a slab of concrete. So it's, in that scenario, the ryegrass is present where we've got anaerobic or wet compacted soils, is where we're seeing the ryegrass issue. So as we build porosity in our soil structure, whether the ryegrass becomes less of an issue, just off those observations. But at the moment, we're still building our porosity in our soil, so it's still a work in progress, and what actions we can take to sort of keep crop production up whilst we're trying to build our soil structure.
Do you want to answer some of the questions that we got emailed?
Yep, I'll just have a quick run through some of that. If you were to be picking between applying biologicals and traces, and you had to pick one over the other, would you be going for on seed or in furrow?
I'll do both.
If you had to pick one or the other?
Oh,
There's like
Well,
4 though, isn't it? Like biologicals, what trace elements on the seed or in the furrow.
But yeah, if you were to be, if you had one option or the other, would you be going for on seed with everything, or would you go for in furrow if limited by what equipment you have on hand, on seed primarily, rather than for early stages if you don't have liquid set up for injecting?
Yeah, well, I think if you don't have a liquid set up, well, then that's your sort of your only choice, really, just to get it on the seed, and at least you got something close and on the plant. Ideally, you do both, get it some on the seed and then inject some in the furrow. And I think, well, we've used liquid inject for a fair while now, and they're not that, you can set them up yourself pretty, pretty easy and not too dear expensive if you get a lot of your own, make your own, get fittings standard just out of the standard shops, and that it can all be done. But I really like it because you have the ability to inject the biology into the soil, with the other fertilizers if you still, granules or whatever. But you can, a lot of the biology you can make yourself and put it in there quite easily and cheap as well, but has a huge payback, I think. Yeah.
We don't have the ability at the moment to put liquid on, so everything's going straight on seed. Like to one day get to the liquid inject, but at the moment we can only go on seed, so that's what we're doing, just everything on.
I suppose on that too, Nicole, if you didn't have the ability to inject it in the furrow and we put it on the seed, then we can also look at coming back as a foliar, with your dissolved urea. We want to talk about, and even some biology, and talk about, if we are going to do some foliar, we can get into sort of some practical tips on dissolving it and adding the humates and things, but how important it is to have things in there that if we're putting biology on the leaf, that we put, whether a fish oil or something, a carbon source to help protect the biology and help it stay on the leaf and things like that. And times of spraying it, how important with herbicides in your tank, then to put it in with biology, like decontaminating and some of those sort of things.
Yeah, go for it.
I do it, mate. Do you want me to?
Yeah.
You're all doing it.
Oh, well, that's one on the foliar nutrition, I suppose, and we can look at. I think it's a real good step, and Joel went through a lot of this, and Graham on being more efficient nutrients. Instead of broadcasting 80 kilos of urea, we could dissolve 15 or 20 kilos. We find a ratio of 25%. So we'd put, what's that? Every 60 litres of water, there's 15 kilos of urea in it. So if we put it out at 80 litres, it's 20 kilos per hectare. And that dissolves even in the middle of our winters pretty easily, that ratios in the water. And there's tables you can Google for solubility at water temperatures and things. But we're, dissolving the urea, adding the humate or the carbon source of humates, and then fish and other biologicals, putting that on the leaf, we're getting, it's, the efficiency, which you can probably touch on compared to broadcasting, we're finding 15 to 20 kilos a hectare. We're getting sort of similar results to 80 kilos broadcast. So there's a huge saving straight away, but also we're finding that the plants, we're lifting the Brix. We measure when it's broadcast, generally the Brix goes down. And then that opens you up for frost issues, insect diseases. When we get that more balanced nutrition out with the dissolved urea, we're getting, bringing all nutrients up, and we SAP test this to check. But then, I suppose on the frost thing, even foliaring, you said the other day, Nicole, with the Pseudomonas syringae and the Pseudomonas, having the Pseudomonas florenzae in the worm and stuff that helps with the frost, and whether you want to touch on any of those things.
Yeah. I mean, I think what you're doing is really fascinating, and I think I often think of foliars as being, we're kind of bypassing the soil processes, but we're then enabling that plant to really set up its microbial community so that it can actually start enhancing or absorbing those nutrients for themselves. And so I think my push for you is to start looking at how refined can you get it and how little do you need, because your soil, I mean, when I went to Grant's place, it just amazed me. We had a penetrometer and it was the middle of summer, and that penetrometer went to the ground in your field, and I've never seen anything like that. Like, it just blew my mind. And it's like, if you are able to do that and start to create aggregation and have a soil system that's starting to work, how much of the nitrogen do you actually need, and how much of these other inputs do we need? And I think, you know, doing your leaf tissue tests and keeping an eye on that stuff is really important. The frost stuff is fascinating, and I mean, there's multiple things obviously involved in frost. One of them is your Briggs. So as we get those sugars and dissolved solids up, your water goes down. So there's less water in that leaf. We find nitrates have a role. So having a nitrate problem in the leaf makes that plant more susceptible to frost. But yeah, these organisms that create those frost nucleuses on the leaf surface is a type of bacteria which you spoke about so beautifully. So your Pseudomonas syringae, which is what makes up about 40% of raindrops as well. So I mean, it's in the atmosphere, it creates rain and these nucleuses. And what eats that is a type of bacteria called Pseudomonas fluorescens, which is fluorescens because it actually does glow in the dark. But it comes out of a worm's butt. So it's one of the reasons that, another reason why I like the worm extract so much is they have this frost-eating organism. And so we've used that in viticulture, which with just phenomenal results, same in avocados. So not only you could do as a foliar, but actually as a soil application, that microbiology will move through the system. You know, it's moving in the xylem. If you're putting it on as a foliar, the biology will actually move through that actual plant and down out through the root system. So, you know, incredibly beneficial. But how do we really start to have a system that's humming? And, you know, if you're doing that seed dressing, that biology will grow as that plant grows up and down. You know, the biology is now inoculating all of those surfaces. So, yeah, I think there's so much that we can do around frost. That Pseudomonas fluorescens, it will protect a plant down to a -6 degree frost event. So, you know, if you're down at -10, you know, oh well, you see frost. But yeah, it's kind of
That's the one that I asked 2 years ago was minus.
Two, isn't it, Nicole?
Sorry.
Sorry.
I was going to say, I had in my notes that was the Pseudomonas fluorescens protecting down to -6 for up to 2 months.
Yeah, after an application. Yeah.
Yeah,
Yeah.
Will
so
anything,
huge,
if you do put some of that out, will anything deactivate it?
Yeah, so your fungicides will deactivate it. It depends, you know, there's limited research really on a lot of the impacts of these different chemicals on types of microbiology. And what's frustrating is we might find, you know, in this case it didn't affect it, and then in this study it did affect it. So I think it's that whole, well, why are we putting a fungicide out on the leaf anyway? You're better to have, you know, a leaf surface that's totally covered with biology. The same as in our gut systems, you know, we need intact microbiome, and if we get a disruption in that, that's where we're allowing diseases to come in. So not using things that are going to be caustic, and that's, you know, where the trace elements, we are buffering those with the fulvics or vermicast or compost or whatever, so that we're not getting the biocide effect, because you think, you know, all those trace elements are biocides, and if we are going out with a compost at 100 grams, you're really not going to be doing a lot of harm. If you are mixing these products, you know, like trying to make your own trace elements, then you're going to add that trace element last. So allow, you know, a compost extract to brew up or feed it or whatever and get good biological population. And then we're using sulfate form trace elements, the biology like your sulfate forms, but allowing that to mix in through, or putting it in dilute, so that you're not kind of impacting and killing the microbiology, because we find it doesn't take much to kill your microbes in your tank. And then in some ways, we're doing stuff where I know we've killed all the microbes and we're still getting a response because those quorum signaling molecules, the proteins, the metabolites, all of that stuff is still intact. So we still get a biological response, even though we're putting living biology on. And you mentioned a worm product before. That isn't necessarily a living product. What it is it's all the proteins and the byproducts of that microbial activity, and that's what signals for the biology in the soil and on the leaf to reproduce. Yeah, so it's just something to consider.
So it's as I said before, the foliar stuff's been probably the biggest learning curve for us around what to apply, and after a couple of years of basic soil tests and also tissue and sap tests, we're starting to see correlations between stuff that is deficient in the soil and is showing up in leaf, and other stuff that's deficient in soil that we're seeing inadequate availability in the plant. And the one that's been standing out for us is calcium, where adequate in calcium in the top 10, 0 to 10, but our pH is a bit low but not too bad. But then in our subsoil, we're desperately deficient in calcium, but we got really alkaline soil at depth, but it's sodic. And we're seeing the correlation between the deficiency in calcium at depth and then also showing up in the plant. So that's become something that we just consider that it's going to be an ongoing maintenance program, that a bit of calcium is just going to be in our foliar thing, but other stuff, it's just year to year, it's constantly variable as to what's actually showing up deficient or adequate in the plant.
Which is kind of interesting because it speaks to your ryegrass conundrum as well. I mean, ryegrass, you probably have a deficiency in sheep or cattle, if I can just park that. But, calcium is made available to the plant through fungal activity. And a lot of cropping places we see are very, very deficient in fungi. And, most of the activity that we do on farm, I mean, everything that we do in modern agriculture is designed to take fungi out, whether or not that's intentional. And so when we're looking at calcium mobility and what's happening with calcium, then it's all about fungi. So what are you doing to drive fungi? How are you feeding them? What are you doing with stubble management, hygiene management? So when you're taking a crop off, are you really looking to feed, your fungi and, putting a little bit of calcium on when you actually take a crop off, and just try and get that system starting to really hum. You got sodic down there below. There's a question here on high aluminium, and that, I don't know if that's a case too, but something to kind of have a look at. It's not. Okay.
No, aluminium's not an issue for us.
Right. So this, there is that question here on what does high aluminium do? So if you suspect that might be the case, dig up plant roots and just have a look. If you've got very shallow rooting systems that they kind of shear off, have you got naked roots? So we're not seeing Rastafarian development. Aluminium's going to impact on all of that. But yeah, what aluminium does is really just cooks those root tips. A lot of you guys in Australia have a major problem with aluminium coming through into your finished grain, into the crops that you're growing. It's in your livestock. Then it's going to impact on other trace elements. And so we're seeing people having difficulties getting trace elements through, let's say, into animals because they're being interfered with high aluminium. So get curious about what's happening with aluminium. And again, all of the stuff that we're talking about is how do we have that massive rhizosheath so those plant roots are able to protect themselves? Because if you have naked, bare roots, you now have basically a hydroponic system that's aluminium's going to come through real fast. And actually, if the world figured out how much aluminium was in some of your products in Australia, you guys might be in some trouble. So I'd get really curious about what's happening with aluminium.
Yeah. Few questions, I suppose, a bit more local to us, Grant, around what's happening over summer and what we're doing, whether perennials are part of the system, whether covers loosen, how we're going about keeping biology fed and rainfall, moisture use. I suppose for us, as I said before, with some of the ryegrass, that a wet drought is more of an issue for us than a dry drought in winter. So for us, using moisture over the summer period to dry profile out isn't actually a problem. It's been a learning curve around establishing in a dry summer, establishing warm season covers, but we're continually learning and adapting to it. But what has your observations been? How have you gone about some of the covers? Have you seen perennials come back, or are you focusing on what you're putting in on annual warm season plants?
Yep. So most of the stuff we do is with annuals because we're cropping. We want it as part of a rotation, but we have started doing some perennials as part of the system as well. The whole thing, because we're in probably a marginal or lower rainfall environment here, and that summer, cover crops or whatever is always a sort of a hot topic on conserving moisture and all that. But I suppose a couple of things on that, I think it's really important to keep cover. So on some of our paddocks, we'll have 100% stripper stubble. So we make sure we maintain that moisture. And if we are going to do some cover cropping, it's knowing your goals and what you're trying to achieve in that paddock. If that paddock has a water infiltration issue or things like that, you might use some taprooted stuff, but keeping in mind that they are going to be detrimental. If you let them go through to flowering and full seed set, they're going to get their taproots down and dry the profile out, which can be a good thing or a negative thing. So it's being mindful of what you do after that. If we are doing that, we will follow it with a multi-species cover crop that then they seem to be able to adapt and grow. If we followed that with a monoculture wheat and we had a dry spring, we're going to have a pretty well failed crop. But if we follow it with a cool season multi-species, they just seem to work, and then we can shut them down before seed set in the spring, and then sort of build moisture again. The other thing we do and look at is so it's sort of species selection and time of termination. If we use cool season stuff through the summer, they don't have a massive, aggressive, deep-rooted root system, so they're not going to be too detrimental to your subsoil moisture. We've done experiments where we've done chemical fallow over the summer versus letting some of these cool season type plants grow, and done soil pits on them just prior to cropping and found subsoil moisture the same. But what the big difference was is where we had a chemical fallow in the stubble, if we had some moisture through the summer, biological activity fires up. They want to feed on a carbon source, which is usually on a living root. If that's not there, they start feeding on your old root systems of stubble. Then you get that rotten stubble syndrome where when you're trying to sow, especially with a disc or any machine, those stubbles are snapping off at the base and can cause real issues at seeding. If we have even just letting the volunteer cereals go for just while there's that little bit of moisture for a couple of weeks before just going out and spraying them, that's enough just to feed the biology and stop that stubble getting sort of chewed off at the base, and can be a big help and gain for later on in the year at sowing time. And we find, even we've got moisture probes and stuff, and you can conserve all that moisture even under heavy stubbles. The top in the dry, our heat and our sun, it's going to evaporate that little bit out of the top 20, 30 centimeters anyway. So if we're going to get a plant, you can either evaporate it or transpire it with a plant, and that way at least we can feed some biology and feed some livestock too.
We don't have moisture probes, so actually testing for what moisture we're really using with the covers, we're not quite sure yet, but I haven't seen a crop following a warm season cover not grow and be able to put it down to moisture. But last, when was it, 2018, when we had that 200 mil, that great big summer rainfall event, we were doing soil tests down to 50 centimeters at that point. And one of the unintended results that we saw from it is anywhere where we had complete ground cover with nothing growing, ground cover with a warm season cover crop growing, bare country with nothing, bare country with warm season, that even down to 50 centimeters, there was two spots across both farms, one that had 250 mil, the other that had 50 mil, that there was moisture right down at the bottom at 50 centimeters. What we don't know is under where we had just cover and nothing growing, whether at 60 centimeters, there was subsoil moisture, or where we had covers below 50 centimeters, whether we drained the profile right out. But to say that was in December, to say that we're conserving that moisture for all that period, I'm not quite sure. But for us here, with the livestock in the system, and when we're trying to build structure and get some porosity into our soils to actually open it up, we're not seeing a massive moisture penalty, but we're being careful on species selection. We're making sure that if we're planting in October, November, December, that we're using millets, low water use plants, and trying to get as long a season varieties as possible, so it avoids going to seed and starting drawing nutrient up out of the roots into the head. So I think there's a big part around that too, knowing what you're wanting to achieve rather than just saying, I'm going to go plant sunflowers because they look nice in a dry summer and then rob yourself of a heap of moisture. But for us, yeah, we haven't seen the moisture penalty, but I know some people have, and I suppose that's where the trick with this whole system is working out what you actually need, what you want to achieve, what's the right time. Is it opportunity rather than every year? But it's given us opportunities in a mixed operation too to continue to feed livestock over summer.
Some of the I think having our observations about what we think is a water penalty, or what is actually allelopathic chemicals, or what is actually a biological response, I think is really good to distinguish, because I think we've come out of a mentality of seeing quite primitive weed pressures in very bacterial dominated soil systems, and seeing where they're growing, and then you'll see less growth where they've been growing in the following season, and then blame it on water, when actually it is it's allelopathic. It is very bacterial dominated, and if we shift that towards more fungal dominated, and I think this is for all cash crops, and then having cover crops that don't have those allelopathic chemicals, then for me it started to pull more questions about have we actually been seeing a water penalty, or is it something else? And I think to look at Grant's place and see how that water is changing through that profile, and if you don't have that hard pan and that limitation on this is as much water as you're going to get anyway, and now suddenly you have that whole profile opening up from the use of biologicals and the cover crops. I think we need to start thinking about water, I think, as a bigger picture.
Yep. And the other thing with that, I'll add, it's great, and we have seen, trialed and experimented, and we've seen detrimental things and had crop failures or not going. We've followed some of these things. So it's all about, it's a good way you learn. But at the end of the day, if you're trying to conserve your subsoil moisture, and in the process lose all your topsoil, you got to ask yourself, I think the important thing is to keep cover on. And if we've got no cover and we get these hot, windy summers, we can lose a lot more than subsoil moisture in the topsoil. The other thing, with these C4 plants, like the corns, probably in this area, there's not much, it's predominantly winter type species. And we've found since we started introducing things like corn and sunflowers, and like Tom said, we don't always let them go through to grain fill and to a big plant, but it's just getting their roots in there. Like if you look at a corn plant in your cropping paddocks versus a wheat plant, the root systems are like 10 times. They've got so much more ability to break up the soil and create that aggregation, which then is a long-term thing, but also they're more mycorrhizal, a lot of these plants. And another way we're getting these into the system is when we're lucky enough like this year to get an early break, we'll sow long season wheats or canolas or whatever, but add, corn, millet, buckwheat with them. They'll grow for 4 to 6 weeks, do a heap of work in the soil, and then they frost out in the winter, and then bang, you've got a winter crop. So these are other ways that we can get them in without them being detrimental to over the summer with our subsoil moisture. We don't have to all plant them in. You can plant them now, like in the autumn, and they'll grow 4 to 6 weeks, and they can do a huge amount.
Yeah, we had stuff. Well, we've planted last year in October through till warm season stuff through till February now, and the February stuff was just leftover seed. And the Feb sown stuff, when moisture started cooling down, moisture was available, has gone absolutely nuts, where the stuff that we planted in December was a very patchy germination, but it's still grown. Obviously it had moisture there, but it sort of moderated its own plant population in that paddock, where seeding rates are virtually the same to the stuff we planted in February. And then the February sown stuff won't quite run up to seed. It's going to have the winter kill. It'll have frost options, grazing options. We're going to put some barley into it post lambing. So yeah, so all sorts of options and mixing it up. But you got to keep. We wouldn't have had, if we didn't have the leftover seed, we wouldn't have been planting this stuff in February. And we've learned something this year. So it's just keeping on adapting and thinking about it.
Any more questions?
Yeah.
I just see one there on how do we shift our soils being more
Yeah.
fungal. Well, that was what, like I said earlier, that adding the humates and the fish in the soil are a good fungi food source, and then minimising or eliminating the things that are detrimental. Obviously, fungicides and tillage and things like that. Plants, like Nicole just said earlier, the legumes, clovers and things really feed into the fungi.
How, if you're in the early transitions, Nicole, and you still got a lot of physical soil constraints and you're trying to build a fungal population, is it going to be a lot of applying fungal products to try and increase population, but it's going to be a battle if you've got physical soil constraints? Do you have to sort of, if you get physical structure improved first, is that going to enhance the ability to have more fungal dominant soils?
It's a chicken and egg.
Yeah.
Because the reason that you've got poor structure is because you don't have fungi active. Because fungi are what give you that aggregate structure that Grant has working so beautifully at his place. And I'm sure you do too. I haven't seen your soil.
We're getting there slowly.
But you've got to look at what are those physical constraints. You know, is it that microbial balance? Is it actually a mineral balance? So is it like high magnesium, high potassium, or is it your management? Is it low organic matter? Is it just you? Are you the problem? So we look at what is causing that constraint. Why have you got that soil structure issue? Why is fungi not active? And typically, you know, when we come into most properties that this is all new and just, you know, running a conventional program, they are very bacterial dominated, have very poor soil aggregate structure. And so, yeah, we kind of go hand in hand in terms of let's feed that fungi, really work on getting them to build aggregates, support the plant so it's really pumping and feeding the fungi, and then have a look at is there a major trace element issue? Maybe you're really low on calcium. What is it that's putting the drag on the system? So you got to really kind of use all your diagnostics on that so you can fast track it so you're not having to feed fungi all the time. Yeah.
So if you were starting from a low base, applying some products, but making sure you're knowing what else you need to change. Yeah.
Yeah. Yeah. Because there's no point not, I mean, I guess it's like human health, you know, we can keep bumbling along and we're diabetic and you want to keep drinking Coke. It's like, you got to figure out what is it that is the underlying causal factor in this. And that's why I think the foliars and the stimulants are great in the transition, but you got to address your big drags, you know, and actually nitrogen, I reckon most operations is not a limitation. Nitrogen is not, it's soil structure. It's poor biological, it's low protozoa and nematodes, so the organisms that are actually helping to cycle. And everyone just reaches for the nitrogen, and actually that then inhibits your natural infixing organisms. So getting really curious about stuff like that. Yeah.
Yep. Well, I think that's spot on because, you know, you see it's quite easy, and I think we should all get out with a shovel and a penetrometer straight after a rainfall event and look at how far that water's infiltrating. And what I see a lot on, even on no-till, but like knife point press wheel paddocks, you get that sort of hard pan layer where the water, when it rains, the water sits there like a sheet underwater and it prunes the roots. And then the lack of oxygen, they're saturated, then it looks like you've got a nitrogen deficiency, but really it's a soil structure problem. And I think with all these foliars, for them to work really well, we need to get that soil structure right. And I think not forgetting the fundamentals. Like we can go for all these type of biological things and foliars and all that, but I think if we get back to the basics of just no-till and keeping cover on paddocks, you know, we found when we went from a knife point press wheel to a disc seeder, that started to, after 3 or 4 years, it really started to improve our subsoil structure, you know, and allowing just not that one pass with a knife point, creating that sort of fine, you know, shattering and breaking up the soil and the finer particles to sort of, you know, work down into the subsoil. Keeping cover, and then, you know, we do controlled traffic as well, just to minimise compaction. And I think some farms that I've seen that just do that really well with a disc seeder, controlled traffic and stubble retention, their soil structures are amazing, even without doing all these other things. And once you get that soil structure right, then all these other things start working really fast and more effectively, I think.
Yeah, totally.
What other questions we got? Any of those other questions, Nicole, that you quickly want to think are topical to run back across?
Oh, the ones that we got emailed originally?
Yeah.
Yeah.
We've got 10 minutes left.
The question, and it's been asked a few times, can wheat and lupins be sown together? I don't see why not. What do you guys think?
Lupins don't seem to tend to like competition at all. Like if it's I suppose that goes both ways. When we used to grow lupins, if you want it as a companion, it's going to do not an awful lot of growth or anything, but whether you get the benefit of it breaking up and making a bit of phosphorus available. But non-mycorrhizal, yeah, I don't know. Haven't tried it. We still got a bit of it on hand from when we grew it a couple of years ago. We've been putting it in covers, but yeah, haven't used use it as a companion. But don't know. Trial.
There's a lot of work on through the North America on, and I've got some of it here, but species, different ones that are planted together can have a positive effect on each other or a yield increase, and there is some species planted together are detrimental to each other. I haven't seen as much. I'd love to see some money and some research done on that here in Australia, but a lot of that we've got to look at out of North America, and so there is a lot of stuff. Generally, you know, legumes can be good companions. You just got to, and we do a lot with canola and wheats, and it's just knowing on what you want to be the dominant plant at the finish when you're harvesting. So whether you're trying to take them both through and harvest the two, or you bomb one out as just as a companion, you know, flag leaf, whatever, and then you know, harvest your main crop. But we find adding some of these companions have got way more benefits than negatives, especially, you know, if through the winter, they don't, even in a low rainfall environment, they don't seem to affect yield. They actually sort of help in some ways, but then it's just being mindful. I have seen them being detrimental when you go into grain fill in the spring with certain species. So it's just knowing that, like tap-rooted species, you probably want them in a dry environment to be your dominant plant coming into spring, because otherwise they're gonna become like a weed if they're in a shallow rooted species type crop. Something to think about. Yeah.
There's a question from the first lot of questions that we got coming through saying, is there at some point when your soil health is functional and working that you could use, what does it say, that you could use a herbicide or pre-emergent, might gain crop performance, but the soil recovery or soil resilience can cope with that shock. So a friend of mine's property was studied quite in depth by New Zealand Ag Research, and what they found was an application of 2,4-D just totally disappeared. So the microbiology, so once you reach a system that's functioning really, really well, the microbiology can break down a lot of these materials into their component parts. The problem is that we don't have this, you know, massively functioning microbial system.
Yeah.
So, you know, I think a lot of the like looking at radiation or petroleum or all sorts of stuff, the role of fungi in actually helping to bioremediate or phytoremediate is just massive. So, you know, if we create a system that starts to become really functional, then the resilience or the impact from using occasional chemicals, you know, and I think that's a pretty good goal, because it's not to say that we don't use any of these chemicals. It's that we don't need to be using these chemicals, and if we do, then you have a system that can recover.
Yeah. It's
They
sort
get
of
more-
something that we sort of battle with here. We know the bad or the harmful ones, and it's where do you find the fine line between, in the early throes when we're trying to increase our soil's health, to take action to get crop establishment or performance, but then not be so detrimental, and where's that fine line? It's not so easy to just say cut it out or go full soil health, but yeah, it's one of those things we're sort of working towards, that we get there one day, that hopefully that there are tools available and that the system can manage it if we need to take action.
Yeah, and I think it's keeping that long-term goal, and also knowing in the short term there might be things that maybe aren't ideal, but we're going to have to do to maintain production. Although, I mean, I was really curious to see a new block that Di and Ian Haggerty had taken on, and they just it was a property that had been using 5 herbicides, multiple fungicides, and they just came in with their worm extracts, and that crop looked terrible. And we had like a field day out there, and there was some consultants and agronomists, and they were all walking around and mumbling and grumbling about, oh, here's all this disease in here, and you really need to apply a fungicide, and this is terrible, and blah blah. At the end of that season, they still harvested exactly the same as what the neighbors were harvesting without using any of those inputs. And so I think for us, it's getting comfortable with the ugly hair stage. You know, we have to go through that. You guys don't understand this, but as a girl, we understand this. You know, like, you got to let it grow out, and it's not going to be pretty for a little bit. And maybe you don't have those fields right next to the road where the neighbors are going to complain about it, but allowing that process to go through without feeling like, oh gosh, I'm going to have to get a fungicide or a pesticide or a herbicide out there, and just allow it. Because we see weeds will go through a cyclical phase, and they're just there and remediating, and maybe they're opening soils up, or your program has been opening soils up, which then releases a lot of bound, let's say, potassium, which is then a signal for your broadleaf weeds, and it's a signal for thistles to germinate. And I see this often in year 2, 3, and 4, as people's properties can get weedier. Leave it alone. It's actually a sign that the system's remediating. And we just have to, there is a bit of that faith and trust in allowing that system to go through it without feeling like, oh gosh, I'm going to have to put this chemical on.
Yeah.
So I think that's where keeping that long-term goal is so important.
You're saying you don't put it on the road for all the neighbours to see. We're on the highway, we can't hide anywhere, so everyone's talking about us.
Well.
But yeah, having that long-term view, sort of occasionally when you got to hang on when stuff looks a bit ugly, or if you're seeing, we've seen a bit more of a change in broadleaf weed pressure than, well, we've seen more of it, but you know, what's that telling us? What's it changing? We don't know, but we're just driving towards that end goal. So some of the stuff we just got to ride out and see what happens, but then take actions where we think it's necessary to make sure that things still like we still have an income.
Mm-hmm.
Yep. I think some of that, what we've been doing on some of it, we've for years had certain paddocks, whether it be highly sodic or soil compaction or different issues, and you're putting all these inputs trying to fix the paddock, you know, lime, chicken, whatever, manures, different things. Oh, the little ones escaped there. And then you're trying to grow these monocultures, you know, wheat will in the cropping rotation, and we're just constantly spending money. And then you've got it, then when you, with all those inputs to remediate these issues, we need to grow these high yields to cover it. So some of these now we just drop out the multi-species, and we find year 1, you know, a lot of these, you know, issues start to disappear. But year 2, we get that compounding effect. Our input costs are so much lower, but then the amount of dry matter we can grow with these things, because they're a lot more water use efficient, a lot less inputs used, and then getting live weight gains using cattle or sheep or whatever and then put that back into crop. And we've found that a very effective way, low risk, low input, but we can actually get a high return or high output from these using the multi-species to rehabilitate, I suppose, land.
Yeah.
Mm-hmm.
Questions here around if we're to build soil carbon, are we going to get more gains out of growing covers or having plants doing that, or applying composts, manures? I suppose that also comes down to, if you're just talking strictly one way or the other, is one way going to do more or the other? But then I suppose there's also your system, what you can work with. Like if you've got livestock, is a cover crop a better option than manure if you're straight cropping manures?
Yeah, so I want you to consider that there's two major soil pathways. One of them is I call it the I'm here for a good time, not a long time honey cycle, which is your compost and your manures and your biochar and any kind of inputs that you're putting on. And it's important, right? So it's respiring, it's changing and fluctuating, it breathes in and breathes out. It's what gets the carbon scientists all upset because it moves a little bit like water. All right, and those cycles are important. They drive some of plant growth, but really what we need to be focusing on is living, growing roots that are pumping carbon deeper down into the soil. And so your job needs to become, how do I support optimal photosynthesis, make sure that my plants are functioning, healthy, have good photosynthetic panels and are pumping carbon deeper down into the soil where it's going to be much more stable. It's going to sit there for a much longer time, and there's really influencing, water holding and nutrient availability. So yeah, there's kind of two distinct parts to that carbon story. I mean, they link in together, but trying to put biochar or compost on as carbon projects, I just don't think has a long-term view. We really need to be focusing on get that pump.
So in that scenario for the compost or manures, you might get the small quick fix of a nutrition out of it, but the long-term carbon stuff is the plant scenario.
Yeah, and the manures or the compost could help lift your bricks, so it could help drive that carbon deeper down, but it's not really your and so you might be using it for that purpose, as opposed to I'm going to put carbon on and blah blah blah, because most of that is respired, like the biology just going to eat it up.
Question here, I suppose, more so for you and I, Simsy, around the system, how to start, where to start. I suppose that's something for for every person to work out for themselves. I don't know how many hours I spent talking to Dad, and both of us were pulling each other's hair out trying to understand it, but it took a lot of discussion how we wanted the system to look, where we could start with what we had on hand. Like for us to do this system, we didn't have the planting equipment, so we had to go buy the disc seeder. But I suppose just got to know what your outcomes are, what your limitations are, and that'll dictate how you start, where you start, and the actions you take. And be flexible. Don't lock yourself in. Like, have no fear of saying I was wrong, and you got to change your course a little bit.
Yeah, I think that's important, and probably to set down some goals like we do you should do, write them down on paper with any area in your life that you're trying to grow and progress. So it's no different with the soil. Write down a goal, how you want its structure to be, its carbon, how you want it to function, and look at things to make that happen. And look at, it takes time because we can't all just go out and buy a disc seeder and a stripper front or whatever, but look at ways that over time when you're upgrading, to slowly change your machinery or whatever to be able to do these things. I think like the disc seeder and the stripper's been a big one for us. It's had some big improvements on soil structure and moisture retention and things like that. So they're sort of tools that we can use. And then on, I suppose, transitioning on inputs, I think the seed dressings are one of the easiest one to get rid of and swap that out. We can, a lot of these things, products, whether it be a fungicide, insecticide, we can, instead of using something that's designed to kill things like pathogens or insects, we can use biological inputs that will design to help things grow, make them healthier, make the plant stronger. So instead of just trying to protect it, put it like a sunscreen, you know, like a protectant on the leaf to stop diseases, let's do a biological thing that'll help it, you know, build its own immune system, put a stronger root system down, lift its sugars. On the, you know, we might, with the seed dressings, then if you are using MAP and DAPs and things like that, I think the carbon, you know, adding that to the fertilizer to help buffer, and then look at even adding in some guano, like blending it in, or things that are going to provide that trickle effect and flow on of nutrients in, and encourage the biology to work, not shut it down. And then eventually, like I think, to transition out of those things and have your soil cycling with the biological stimulants and nutrients is where we want to get to, I think.
Yeah, I think the biggest thing is you've just got to know set out what you want it to look like, and then pretty quickly you get a priority list, what you do need to change, what you don't, what you got on hand. But yeah, it's not a slow process, and it's forever evolving.
Yeah.
And for us here, we had to go to the disc systems allowed us to establish covers in a very dry, moisture limited environment and maintaining the cover, but in higher rainfall areas, the disc and complete stubble retention and residue handling like we got, it mightn't be the first point of call that you had to go to, but for us, the disc opened up more opportunities, so that's the first avenue that we took.
We this year's last summer we had a fire go through a strip of one of our paddocks in a strip of stubble. So it was just sort of your worst nightmare when you see a fire. We weren't even in that paddock harvesting and looked over and saw that going. This year we've got I've sown winter canola in the strip of stubble early with millet, corn and buckwheat and things like that. But just where that strip of fire went through to the soil that we've had in this system for a long time, so it's a nice sort of structure and good cover on it. It's unbelievable, chalk and cheese, the difference in what we've got growing on where the fire went through and where it didn't. So we've got to be mindful of some of these things. They can have a huge negative effect on our biology and function of the soil. It looks like it's been eroded and windswept, and it's just terrible really. But off in where we've kept, where it didn't burn and got plenty of stubble, it's like a mulching effect. That soil just keeps getting softer and softer.
Yeah, and I suppose that's a early days throw is you might see a little bit of moisture benefit or something out of the stubble retention, but you mightn't see, it takes time for that effect to eventually show up as to what that long-term retention of ground cover means. And for you've had an unintended trial where someone's burnt it and it's like, okay, well, we've seen a result in one hit of what removing it does.
Yeah.
Someone point out that there is a chat box that we've all been replying into, so you might be curious at seeing what some of the questions have been asked and answered so far. So that's just on the side.
What else is showing up in these Q&As at the moment? Have you ever had any disasters to learn from? Plenty. I suppose for us, our biggest learning curve was dropping off the high analysis FERT and how little we could go in a foliar program. So some of our crops haven't looked fantastic during the year, it's been slow, but we've still harvested reasonable crops, but it's been a learning curve over the couple of years of what we need to bring in, how we need to keep adapting. For us, probably actually ryegrass, some paddocks where we've said, let's go for soil health, take some harder chemicals out of the system that would control ryegrass more. We've had crops run away where we've just had to turn to livestock to harvest income off that paddock. So yeah, there's been plenty of disasters, but make sure you learn from them.
Yeah, I think it's important not just to, you got to keep doing what you know works. Don't make, you go out and do the whole farm and it's the first time you do it. Sometimes these things take a couple of years to adapt and to change. So you might want to transition in stages a bit, so so that you can slowly bring things in, and if something goes wrong, you're not having it go wrong on the whole farm. But don't be afraid. And I suppose that's where this sort of like VicNoTill and these sort of groups and sort of events really help, because if you're going to try something, there's generally someone out there that's done it and maybe can share ways to help minimize the negative experiences, I suppose. And that's where I really encourage everyone to be a member. Like it's to be a member of VicNoTill, it's not a huge cost over the grand scheme of what you do, but there's a huge benefit that you can get out of it, just through the networking and the sharing of, farmers helping farmers. So.
Yeah. No one will ever tell you the right way or the wrong way to run your farm, but there's always an idea somewhere that might steer you in a certain direction to improve or adapt.
Yeah, and I think for me, some of our early disasters were, going to see one speaker who had an idea and going, I'm just going to do what that speaker's advocating. And so we pulled our, fertilizer inputs, started making compost teas, and they were really good compost teas. But we totally crashed that mineral system. Ended up with some real phosphate issues, some trace element issues. We had livestock, just half a dozen die, but that was bad enough for us. And it was just a really steep learning curve. And I think this is where it comes down to, having groups like VicNoTill, discussion groups that you can share maybe some of that experimentation between each other, because there are minefields in this, and particularly I think if you're pulling the rug out too quick and the system hasn't rebooted, we can have issues.
Yeah. For us, we got 3 paddocks that we've set aside. One that's staying in crop rotation, where we're continually planting warm season covers in there. Some other grazing paddocks where we've just no herbicides, no fertilizers, just seeing what happens with just continual plant growth. And from those paddocks where we're still applying broad principles across the whole farm, we're taking learnings out of each of those paddocks and applying them in different scenarios in different paddocks. So we've got broad scale trials of whole paddocks happening, but then taking learnings out of that and applying it elsewhere on farm. There's just, seeing the chat here, a question came up around the profit and loss of insecticides and when it's required. For me, all I did was, I think GRDC, there's a few locally here in Australia, there's a couple of websites that just basically have thresholds for certain pests. And that's where my starting point was when we're looking at reducing insecticides, was just going on there, finding what industry recommendations were for thresholds. And sometimes I was happy to exceed those threshold numbers because I'd see beneficials present and starting to be active, but there's still some industry stuff out there saying these are what we consider economic threshold levels, and then find your own comfort level as to how much pressure you're happy to see. It's a good starting point. Some of the thresholds that are actually on those websites, you wonder why an agronomist would recommend putting an insecticide out, because it's well below threshold levels. So there is information out there as to a profit and loss scenario that you can gauge for insecticides.
I also want to kind of pull it back to insects.
I remember years ago, a lot of the times the recommendations would come in just to put an insecticide in just because you're going over the paddock anyway, and it's cheap. Then, that's kind of a dangerous and a just in case sort of thing. You're wiping out all your predators and beneficials, and the pests are pests because they can reproduce rapidly and they come back quicker and harder. So, years ago, we might have just, when we get pressure, it's off the fence lines coming in. So we might have just done one lap around with the insecticide on a heap of paddocks and then after time we just ditched it all together. But now we'll do, you might do a sap test of a plant, see if there's anything real out of whack. And if it is, generally liquid calcium's great because it'll lift sugars, and then putting fish and humates and different things in, because there's, Nicole can touch on this, there's like a strong correlation between a higher Brix becomes less desirable for insects. So we just focus on getting that up, through nutrition and biology, and then all those symptoms, diseases, insects, or frost, stem frost, all those things just start to disappear, and to a point where a lot of the time we don't even worry about them anymore, like insects or diseases. It's just not something that we look at, but we just got to make sure, so whether you want to touch on importance of getting the Brix and, minerals that are low or nutrition that is out of balance, to getting those things, addressing them.
Yeah, and so I would get not only a refractometer, but a SAP pH meter. And what we find is there's definitely a correlation, obviously, with Brix and with pH to insect pressures. So if you're seeing that SAP drop right down, then using a nutritional spray instead to be the deterrent against insects rather than an insecticide. So we've done this on a lot of high value crops. We are actually applying nutritional foliars instead of an insecticide. So insects can't digest complex amino acids. So if we have nitrates in the system, if we have what Jerry Brunetti used to call the funny proteins, if you have those kind of floating around in the system, then that's ringing the dinner bell for insects. So how we complex that is with some of these carbon-based products, the humics, the fulvics. I'm a big fan of milk, so using milk powder, for instance. So you got a bit of calcium, you got some sugar. It'll help complex some of those free nitrogens in the system and actually change the pH and the sap of the plant. So using a sap meter can give you some cool information.
Yeah, right. Interesting.
Yeah, milk sort of high that calcium phosphorus thing in that too. A strong component of it, which I think is obviously good for photosynthesis and sugar production. And I've done experiments at home tipping milk powder on tomato plants, and the yield response you get's amazing, and the health of the plant. I was just trying to look at when you're saying the pHs, even pH in the animals' urine and different things like that, and in plants where you want them to be for to be healthy and disease and things like that.
Yeah, it's super fun, and I know quite a few dairy farmers that are using this. So using your pH strips to test sparples, so you can just buy like these packets of pH strips, and you can actually get saliva from those cows, and that saliva should actually be alkaline because the rumen is a bicarbonate environment. So our pH in our mouths should be between 6.8 to just over alkali, whereas cattle are around 7.8 to 8.4. And what you find is if those animals start to get more acidic, you start towards acidosis, using those pH strips can actually give you an early warning before you show clinical signs of mastitis or whatever. So that it's quite fun to do. With the pH in your sap, it should be 6.4 of any plant. And I was on a vineyard that had a sap pH of 3.4 in their vineyards, in their vines. And I said to them, do you have fungal problems? And he's like, no, our fungicide program's working really well. I'm like, oh, okay. Oh, but we're pulling 15% out a year due to virus. So, having a sap pH like that, I mean, it's nearly battery acid that's running through those plants. You've got a major nutritional problem. And what he discovered was applying the fungicides drop that pH even more. So the fungicides were actually creating the environment for more fungal diseases, and down we go. So being able to use pH like that to see, oh, wow, actually what I'm doing is actually making the problem worse is a valuable tool too.
Mm. Yeah. Just scroll through.
There's I don't know, you can use, I think it's, what was that?
You're right.
Mine. Just saying, with your bricks and all that, you know, looking in the morning versus at night to see it going up and down, you know, before or after you apply a herbicide or any sort of foliar or different things to your paddock, it's a good tool that we can just have in our pocket and
Yep.
check. And there's ways you can see if your boron's, you know, potassium, calcium, we can sort of pick up on some of these deficiencies or whatever in the plant by just using a Brix meter. So, and that's all been, you know, we've had that in articles in the VicNoTill magazine. We've done a whole section on that, and I think it's just a handy tool we can all have.
Yeah.
There's another one too that we played around with recently. Remember in New Zealand, that biometer?
Yeah. The biometer. It kind of gave us a score for, you know, is this poor soil or good soil? I mean, I think you can visually see that for yourself. I don't think you need a fancy scorecard to tell you that. Dig a hole, you know.
Yeah.
I don't know. A number, I guess, for people that need numbers, but
So your undies.
yeah, put your undies in the ground and how quickly do they disappear? Okay, there's going to be lots of we're not going to be able to answer in the next 10 minutes.
Yeah.
It's question, no risk of sending a soil anaerobic from using milk. You're not going to be putting that much on. So on pasture, we've been using about 40 litres or 4 gallons. We've used up to 75 litres, and we have used it in viticulture for control of like powdery mildew. Works really well on a lot of those surface kind of rusts and those soft diseases. Really don't like milk, which is quite fun. So no, you will send it anaerobic if you're going to pour milk on, like these guys that are dumping milk at the moment. That's not good. We're talking about small amounts.
Yeah.
I remember there were some earlier questions on teas and extracts. Do you want to pros and cons of any of those? The compost teas, compost extracts?
Yeah, so I used to do commercial compost teas, and I just found that there's so many more things that can go wrong with a compost tea compared to an extract. You've really got to be very diligent with a lot of like your things like your dissolved oxygen, what kind of foods you're feeding, who are you actually breeding up when you're breeding up a compost tea. A lot of bacteria obviously go nuts in a compost tea, but less so like maybe your fungi or your protozoa or nematodes. So I ended up going right away from compost teas. Now I use slurries. I just love a good slurry. So the slurries are, you're putting on more compost than you would in a tea. Like say in a tea, you might put on 0.5 kilo per hectare. In a slurry, you might put on 20 kilos. But you're basically sending out the house as well as all the metabolites. I think you get better survivability. You just need equipment that can spray a slurry. And then you've got the extracts, which is what Grant's doing. So, you know, blasting, using air or water to actually extract or take that nutrient off. And someone asked the question before about how do you know about your vermicast or worm extract? You're looking for color. So when Grant turns on his machine and it just turns to this beautiful dark chocolate brown, that's what you're looking for. If you're running water through, say, compost or vermicast, you want to run that water and keep that lovely dark color. And then if it started to go paler, or it started to go kind of a golden color, then that's when you turn your water off because you've extracted all those humic substances off at that point. So in extract, we're really looking at how do we get all the metabolites off that surface. So lots of water pressure, air pressure, depending on the types of equipment that you're using. But I find extract a lot easier in a big commercial system like these guys are running. So you can put that through a lot of your gear a lot easier than a slurry. Slurry, you end up putting out a lot more water. I don't know where Grant, but yeah, did you want to add something, Tom?
I was around the brewing process, and I think the Haggerty's, like, they just feel that's what they fill their sprayer with for their application process, and then to activate it is just adding worm leachate that's going in the tank out the paddock. Like, that's mass scale, making it as simple as they can for their process. So you can make it as complicated or as simple as you like the system to be.
Yeah. And I think we humans like to really overcomplicate things, and it's like, no, simple, less is more.
Yeah.
Yeah.
And for us, the simple, like to go and set it up mightn't be a costly thing, but at the moment for us, the practical is to be buying off product shelves. Like that's what we found to be an efficient method at the moment, and that might change.
Yeah.
Am I in?
Yeah, you're back
here?
on.
So the kids just turned on Netflix, and I think the internet just overloaded. What was interesting with the extracts and that, after being in New Zealand, I got one of those microbiome meters, and we had a workshop here just before the corona stuff. And it was actually interesting to test, to use on site to see the effectiveness or how we're doing the extracts. And I ran the extractor, and we did a test within 10 minutes, and you could even just visually tell the water was quite, hadn't gone that chocolate dark color yet. But we grabbed it, tested it, and it was quite low. But then we kept running the extractor for, whatever, a couple of hours. And then we tested it again later, and the same, without doing anything, just giving it more time, and it was through the roof. So I think,
that would be value. That would be of value. I would use it for that. That's cool.
Yeah. But no, it was handy just to sort of see that. But I don't know, I dropped out there whether you said, but the extracts can have a more diverse range of microbes, because like you said, I think you'll touch it, like the teas, you can sort of, that can enhance one specific species, the aerobic ones or whatever, but the extracts are more diverse, I suppose.
Yeah, definitely. Yeah.
Less fiddling.
If you're doing compost tea, you want to be relatively competent with a microscope. The amount of people I've met that are making these anaerobic, bacterially dominated nastiness in their compost teas, and they're not looking at it in the microscope and then applying it. And it's like, well, no wonder you're having more disease problems and loss of soil structure and all sorts of stuff. So, I think compost teas can be used well with people that really, you've got to understand a system a lot more than a slurry or an extract. And it's not saying that compost teas are bad. I just think you need to know a lot more about what you're doing.
Yeah.
All right, we've got 3 more minutes.
Yeah. Anything else that you think's worth covering or bringing up, Nicole? Just briefly, or closing it?
I've been trying to type people's. Yeah, I think maybe if we just sum stuff up.
Yeah. I suppose from a practical standpoint, my comments are that it's know what you want to achieve. There's knowledge out there about people have tried different things, so it's easy to find a bit of knowledge. But the easiest thing to do is to start and know what you want the system to look like, and then source the information and techniques how it might work elsewhere. But it is a learning curve, and to know the outcome you want to achieve will go a long way to getting some success and believing in it. If you don't believe in it, not going to happen.
No, which I think just comes back to the mindset issue that we have. If you think you can or you think you can't, you're right. And I've seen a lot of people sabotage their success by just thinking it's not going to work anyway, and so they get their timings wrong. They don't check their brews, or they just mess about with products or allow stuff to get too hot. Like there's so many things that you can do to really, when you're dealing with a living system, to kill your system.
And I think for us, some of the biggest learning has come because we knew what we were trying to achieve. So if something did work or didn't work, we can, when we didn't get the outcome we wanted, you can analyze it a lot more rather than saying someone said to do this, I just did it. If you know why you wanted to achieve it, then you can analyze what went wrong and you can improve a lot quicker.
Yeah.
So
we recorded this call, right? So that this will be available to people.
First time doing it, but hopefully it's been recorded correctly.
I recorded on my side just in case there is a problem.
Okay.
Yeah.
Dimsy?
Oh, no, I was just going to put in my little bit here, but yeah, just sort of thanks for asking me on here, and no longer on the board, but know what goes into this. And I think it's great you guys, VicNoTill, have done this, in these tough times, and we can't have the more, you know, the model they've been using with the workshops and the conferences, and just sort of asking everyone here to keep supporting, you know, by being a member, support the sponsors, you know, that help put this together. You know, these guys, everyone's doing it in different times at the moment. So I know, yeah, VicNoTill really values the contribution to all the sponsors and the members, and yeah, just keep supporting each other, and that's how we all can grow with our soils and businesses and things like that.
Yeah, really appreciate everyone's questions and participation. It's been a lot of fun, and I think, you know, if you're not sure where to start, just start somewhere small. Just start with a trial. Just, you know, just do it, right? Don't, you know, the greatest regret I see with people is that they wish they'd done more earlier. You know, they hear an idea and you sit on it for a few years, and it's like, just try something. Put a little bit of humic in. I don't know.
Yeah.
Try a bit of a worm extract. Try buffering some of your trace elements or whatever. Start doing testing, looking at what's happening with your leaf samples. You know, are your plants actually have an excess of nitrogen? Because we see that more than not enough nitrogen. So, you know, just start building up your knowledge base. And yeah, I really appreciate all your questions. So thanks, guys.
So before
read my book.
before we get to close it again, this is the first webinar that we've done. Hopefully it's gone well. We've had over 240 people tune in, which is unbelievable for the first one. Thank you to the Australian Government Land Care Programs, Wimmera Catchment Management Authority for their support, and VicNoTill's Platinum sponsors, Hybrid Ag and Lorico. Nicole and Grant, thanks for taking the time to sit on here, and I'm sure we've given people a bit of a headache, but also given them some ideas to head down. So thank you for taking the time.
My pleasure, guys.
All right.
Thanks for being
Very
on.
good.
I'm going to go back to the snow. Back to the snow.
See you guys.
See ya.
Yeah.